Over the past 3,500 years, metals have been utilized in medical applications to treat illness throughout many countries and cultures. In recent medical research development, ruthenium has been the focus for a variety of treatment applications, including immunosuppressants, cancer treatments, dental alloys, antibiotics, antimicrobials, and septic shock treatments. Ruthenium, element number 44, is a second-row transition metal, and its chemical properties allow it to be a very useful therapeutic tool in pharmaceuticals. Three of the most important properties of ruthenium consist of its rate of ligand exchange in biological systems, its propensity to adopt multiple stable oxidation states at physiological conditions, and its ability to mimic iron binding to certain biological molecules. Ligand exchange chemistry is the key to cancer-fighting metallopharmaceuticals, and certain platinum 2+ compounds have been very successful. However, inorganic chemists in the pharmaceutical industry have focused lots of research on platinum 2 substitutes due to its inability to fight some high social incidence types of cancer and its toxic side effects. Ruthenium 2 compounds NAMI-A and KP1019 effectively address these problems. Both NAMI and NAMI-A share the same pseudo-octahedral ruthenium 3 center, surrounded by 4 chloride ligands in the equatorial plane. One S-bonded DMSO and one imidazole occupied the 2 axial positions. The geometry of the ruthenium 3 in KP1019 is similar to that of NAMI-A. Much like cisplatin, NAMI-A becomes active upon hydrolysis. At pH 7.4, a chloride undergoes ligand exchange with a water molecule, which neutralizes the molecule's -1 charge. Upon lower pH, imidazole is cleaved and replaced with water. Once the ruthenium compounds have replaced the chlorine atoms with water, they undergo a ligand exchange with Lewis base sites on DNA residues. Both compounds show overwhelming affinity for guanine, and their irreversible binding causes a conformational change in DNA's structure, preventing RNA synthesis. However, looking at the crystal field diagrams of both the ruthenium compounds and cisplatin, the ruthenium compounds have markedly less crystal field stabilization energy, making them much more labile. This difference in ligand exchange rate between cisplatin and ruthenium compounds is thought to be the source of much of the mechanism of action difference between the metallopharmaceuticals. Even though all of these compounds readily bind to tumor DNA, they both produce several species that are capable of reacting with a variety of biological components due to ruthenium's unique properties. It has been observed that NAMI-A binds to proteins on the tumor cell membrane, leading to decreased cancer invasiveness and lower cytosolic toxicity, while KP1019 does not and causes apoptosis. Another very important use for ruthenium in medicine lies in its antimicrobial applications as a drug target for malaria. Quinolines have been the most successful and widely used treatment for malaria in recent years. The major mechanism of action of chloroquine is to prevent the formation of hemazoin and causes malarial cells to lyse. However, resistance to this drug is caused by a mutation from a charged lysine amino acid to a threonine in the PFCRT transmembrane transporter localized in the digestive vacuole, causing chloroquine to bind to PFCRT, rendering the drug inactive. A ruthenium 2 chloroquine complex presents a viable solution to this problem. As lipophilicity of the compound increases, the PFCRT transmembrane transporter decreases resistance to extrusion. In vitro, the ruthenium chloroquine complex undergoes a ligand exchange between equatorial chlorides and water molecules. The subsequent compound is a ruthenium 2 hexacoordinated compound that is stable at physiological conditions. Axial chloroquine ligands then bind to hemazoin crystals and impede addition of solute. The presence of ruthenium altogether allows for raised lipid philicity and small conformational changes to chloroquine that prevent PFCRT binding and promote localization around water-lipid interface regions within the digestive vacuole of the parasite. All in all, ruthenium provides many useful applications to modern medical treatment, from dental alloys to chemotherapy. Its unique versatility allows it to be applied in many aspects of chemistry and biology, whether it be for vital redox reactions or simple structural rearrangement.